Protein synthesis consists of two parts, transcription and translation. The production of new proteins inside of a cell is referred to as protein synthesis and occurs in the cytoplasm. Transcription is the first step in protein synthesis. For proteins to be made properly, the genetic code for how to build the protein must be followed. DNA contains the genetic code to build the proteins of the cells. However, DNA is located in the nucleus, not the cytoplasm. Considering the code to build proteins is located in the nucleus, yet protein synthesis occurs in the cytoplasm, the question arises, how are proteins able to be built in accordance with the genetic code? Transcription is how the cell is able to solve this problem. When a protein is to be built, the first step is to express the genes for the protein. This means the genes, which are the parts of the DNA that code for the protein, become exposed and copied. When a gene is copied, the copy consists of a single-stranded mRNA, not double-stranded DNA. The process of copying a gene and producing single-stranded mRNA is transcription. The single strand of mRNA then leaves the nucleus and arrives in the cytoplasm. Thus, transcription of a gene allows the protein to be built in the cytoplasm, even though the genes for the protein are contained in the nucleus. The protein is synthesized during the step of translation. Proteins are made of amino acids. While different proteins take on numerous different shapes and can be large or small, all proteins start out as a single chain of amino acids. Inserting the wrong amino acids or too few amino acids would mean incorrectly building a protein, which can have disastrous consequences on the functions of the cell. This is why it is so important to ensure the genetic code to build the protein is followed. This is the very problem that translation solves. The mRNA contains the code of which amino acids are to be used and what order they are to go in when building the protein. During translation, mRNA binds with ribosomes. The ribosomes allow for amino acids to be added together in a chain only by following the code provided by the mRNA. Thus, translating the mRNA ensures the amino acids build the protein in accordance with the genetic code for the protein. To review, transcription is the process of copying the code to build a protein so the code can be present in the cytoplasm. This is accomplished by producing an mRNA copy of the genes for the protein. Translation is the process of translating the mRNA, making sure the amino acids joining together to build the protein follow the genetic code for which amino acids and in which order the amino acids should join together to build the protein.